All right, so nucleic acids are the last of our four biopolymers. Nucleic acids are composed of carbon, hydrogen, oxygen, nitrogen, and phosphorus. They are the biggest macromolecules, largest molecules in the body. They're polymers made up of a monomer called nucleotides, which is composed of a nitrogen base, a pentose sugar, and a phosphate group. There are two groups of nucleic acids are deoxyribonucleic acid and ribonucleic acid, commonly referred to as DNA and RNA. DNA holds a genetic blueprint for all proteins. It's the instructions to put proteins together. It has the primary sequence of proteins encoded into it. It's a double-stranded helical molecule called the double helix structure located within the nucleus. And the nucleotides contain the deoxyribose sugar, the phospho group, and one of four nitrogen bases, an adenine, guanine, cytosine, or thymine. Those come in two different forms, the purines, which would be adenine and guanine, and the pyrimidines, which would be cytosine and thymine. And a good way to remember the nucleic acids is to remember, for DNA, the phrase pure silver tag C. That will allow you to remember everything you probably will ever need to know about DNA. The purines are adenine and guanine. Ag is the chemical symbol for silver. Pyrimidines would be the other two. And tag stands for T always binds with A, and G always binds with C. So here that's spelled out. So complementary base rules. A pairs with T. If you've got an A on one strand, you got a T on the other. G always pairs with C, forming this double helical structure. G, C, C, A, G, A, T, T, A. The nucleotides bind together. RNA is the kind of the link between proteins and DNA. Its structure is quite different from DNA, even though chemically it's not much different. RNA exists as a single-stranded linear molecule. Most of its work is done outside the nucleus. DNA never leaves the nucleus. It has a ribose sugar, not a deoxyribose sugar, so it's got one more oxygen. And the bases, instead of a thymine, a T, you have a U, a uracil. But even that uracil is just a difference of a single methane group. So there are three varieties of RNA that we need to deal with that carry out the instructions that DNA holds for building proteins. They are messenger RNA, transfer RNA, and ribosomal RNA.
DNA and RNA are nucleic acids, polymers made of subunits called nucleotides. One difference between DNA and RNA is the type of sugar their nucleotides contain. DNA contains the sugar deoxyribose, while RNA contains the sugar ribose. Ribose has one more oxygen atom than deoxyribose. DNA and RNA are each composed of four different nucleotides, which differ in their nitrogenous bases. Three of the four bases are the same in DNA and RNA: adenine, guanine, and cytosine. The fourth base in DNA is thymine. In RNA, it is uracil. The nitrogenous bases guanine and adenine each have two linked rings of atoms. They are called purines. Cytosine, thymine, and uracil each have a single ring, and these three bases are called pyrimidines. For convenience, the carbon atoms in a nucleotide sugars are numbered, beginning with the carbon atom bonded to the nitrogenous base, moving around the ring, and up to the carbon that is bonded to the phosphate group. The 1' carbon is bonded to the nitrogenous base, the 3' carbon to the next nucleotide, and the 5' carbon to the phosphate group. DNA and RNA are polynucleotides, long chains of nucleotides. Polynucleotides are always assembled in the 5' to 3' direction. A covalent bond forms between the carbon at the 3' position of a nucleotide and the phosphate group at the 5' position of the next nucleotide.
Lost our audio, but the two DNA chains or strands are oriented in opposite directions and held together by hydrogen bonds between the nitrogen spaces on opposite strands. Because of their sizes, shapes, and arrangements of polar groups, the DNA bases form complementary pairs. Adenine pairs with thymine and cytosine pairs with guanine. Two polynucleotides in DNA wind around each other to form the familiar double helix. The bases can be in any order, like the letters of the alphabet. However, the base sequence is significant. Sequences of bases called genes encode the instructions for structural and functional proteins of an organism. That must be the end. All right, one more nucleotide we have to be aware of. That is ATP. Chemical energy used in the cell is used by removing a phosphate from an ATP. ATP stands for adenosine triphosphate. Adenosine triphosphate, ATP, directly powers chemical reactions in the cell, offers an immediate usable energy needed by things in the cell. ATP is an adenine-containing RNA nucleotide with all 3 phosphates attached to it. So we've got adenine, ribose, excuse me, and 3 phosphates. The 2 outer phosphates have high energy bonds, and we use the 3rd one all the time for energy. Sometimes we can use the 2nd one for energy. With that 3rd one out would be the terminal phosphate group. If you take that phosphate group off, you have ATP becomes ADP, or adenosine triphosphate. And if we use that 2nd phosphate, the ADP can become AMP, or adenosine monophosphate. We'll see that as a 2nd messenger in chapters coming up. So ATP to ADP releases the phosphate, inorganic now because it's not attached to carbon, and energy. And that energy is used to move something in the cell, maybe an active transporter, moving something from inside the cell to outside. So transporters, muscle contraction, all chemical work is going to involve ATP being converted to ADP, and then using the energy from carbohydrates to put the phosphate back on ADP to regenerate ATP. All right, covers